Chapter 15 is all about different lab techniques and, I guess, physical exam techniques to diagnose infections. So I'm going to give you an overview first of these different laboratory techniques, and then we're going to kind of go into detail with some of these different lab techniques so you understand what's actually being performed in the lab, sort of assuming you're coming from the position of nurses, taking the samples from the patients, sending them down to the lab. A lot of times doctors and nurses don't really know or understand what's going on in the lab to get that diagnosis, which can lead to a lot of frustration on both ends because doctors and nurses can call the labs like demanding answers, but because they don't understand the steps that need to be taken to do the tests. So some of these different methods are you can do direct testing of a sample using microscopy, or using immunologic techniques, or even doing genetic testing. But a lot of times with microbes and microbial infections, you have to actually cultivate the organism, grow it, culture it, in order to be able to truly isolate it and identify what the pathogen is. So depending on which tests you can do, if you can do direct testing, you can get a quick result, but if you have to cultivate it in order to isolate it, then it's going to take potentially a couple of days or even weeks depending on the organism. All right, so the overview of the different types of identification techniques. There are phenotypic, genotypic, and immunologic assays, or the three sort of categories of assays. So phenotypic is looking, basically think of like looking at the traits of the organism. So we can do this through microscopy, different staining techniques. We can also look at, do different biochemical tests. So like using different types of differential media, for example, to look for color changes that give us clues about, and there's other tests that we'll talk about, enzymatic tests to look for enzyme function. We can do genotypic tests, looking at the genotype, the DNA or RNA sequences, in order to identify the organism. This is more expensive. A lot of smaller hospitals don't have this equipment or technology on hand. They have to send out the labs to a genetic testing facility, so that does increase the amount of time that it takes to get results, but it's very accurate. And then immunologic assays often can happen on site, but these require sort of kits that have been pre-made. So not every type of microbe has an immunologic assay that can be done. Really, there's only a handful that have these assays, but they're really great because they're very specific and they're very sensitive, so they can detect even very small amounts of a microbe. So for the microbes that we have immunologic assays available, they're kind of a gold standard. Okay, so let's start with these phenotypic methods. The first thing we could do is look at the microbe under the microscope. Just some simple staining so we can see what is the shape, or some gram staining is very standard procedure to do some gram staining, determine whether it's gram positive or gram negative, and then also to look at the morphology. What's the shape of the cell? Is it coccus, bacillus, something else, vibrio, spirochete? Sometimes you might be able to even see other appendages like flagella or endospores, but generally just knowing whether it's gram positive or gram negative can be give the doctors a leg up in terms of which antibiotics to prescribe, and also fast track to figuring out the diagnosis. We can also we can grow the microbes on a petri dish, on solid agar, and we can look at the colony morphology. We can grow them on selective and differential media and look at the different colors. We can look at the size and shape of the colonies. They're different between different bacteria sometimes. So sometimes that macroscopic morphology, how a colony looks versus how the bacterial cells themselves look, can also give us more clues. We can also look at different biochemical characteristics. What enzymes, what sugars can the bacteria ferment? And we already did that with some experiments in lab with selective and differential culture media. We can sometimes identify by different sugar fermentation. So there are these cool kits. they're really expensive, so I could never get them for this class, but it's called an enterotube, as well. It's one of the names of these products, where it's basically like a bunch of small little petri dish wells, like a dozen of them, and you take some bacteria on the tip of a needle and you stick it through the whole thing and inoculate all of those cultures, and then they each have different sugars and different dyes, so depending on which sugars the bacteria can ferment, certain ones will change color, and then you get sort of like this color code, and you look it up in this manual, and it will basically give you a diagnosis, tell you what bacteria it is or what family of bacteria it is. So it's pretty cool because it's basically like inoculating 12 petri dishes at once and sort of reading them all at once. So those are pretty neat, and they're very I don't know, convenient. It's a convenient way to assay several sugars or several biochemical characteristics at once. You can also do chemical analysis of the makeup of the bacteria, but this also takes some fancy chemistry that's not always available. Genotypic methods involve looking at the genetics, the DNA of the microbe or RNA, and doing some kind of genetic assessment. So there's some advantages and disadvantages of this technique. So the advantages are that you can identify microbes that are non-culturable. Remember from that article that you read about tioxobactin and discovery of new antibiotics that we really only are able to culture probably about maybe 1% of bacteria on this planet. So 99% of the microbes out there are unexplored territory and are uncultured. We can't culture them, we can't really study them. And so if that's true of a pathogen, sometimes there are pathogens that can't live outside the host. And if we can't figure out how to culture it in a lab, we'll never be able to grow it, to identify it. So looking for genetic signatures is a way to identify bacteria or viruses or other microbes that we just can't grow in the lab. Genotypic testing DNA with the advances in DNA sequencing technology, we can get those results in a couple of hours if you have that machinery on hand. If you have to send it out, you probably have to wait a little bit longer, a few more hours for the distant lab to get the sample and run the test. But it's still quicker than culturing, because with culturing, you get growth in 24 hours, but then you still have to do tests on that, because growth doesn't necessarily mean identification. Whereas genotype tells you exactly what it is. It's very specific. But it is still kind of expensive. That's a disadvantage. Not all labs have the technology on hand, or maybe even the cost in their budget to be able to ship samples out constantly. The other thing is that although it's nice that you can identify non-culturable bacteria, you also will identify dead bacteria. And so if there's a bacteria that happens to, some dead bacteria in a wound that you sample, and they're not causing the infection, they just happen to be there and they're dead, you would get results for that, and you might mistake them as the cause of the infection when really they're just some dead bacterial cells there. So also not perfect technique. Immunologic methods. These are really useful. They're very sensitive and very specific, and you don't even necessarily have to find the microbe. Sometimes you just test what antibodies the patient is making to see what they've been exposed to. It's usually really easy, really quick. It's like a kit that's been prepared. But the disadvantage is that they're only available for a handful of pathogens. So the ones we have it for, it's awesome, but for most bugs, we don't actually have immunologic assays for. All right, so before we can do any of these tests out in the lab, you have to collect a specimen. And usually the frontline healthcare professionals, nurses, and doctors are the ones who are doing this collecting. So for a lot of you students, this would be your part of the job. So you might see a tube like this where it has a swab and it has a little culture media at the bottom, and when you open it up, it sort of lyses that culture medium. You take your, you swab the patient's sample, whether it be skin, throat, nose, whatever, and then you stick the swab back into the little media and you send it down to the lab with identification and testing specificities. All right, so there's lots of different sites that can be sampled. Throat and nasopharyngeal samples is what's been going on with the COVID-19, the SARS-CoV-2 coronavirus sampling has been throat and nasopharyngeal samples. Urine samples, usually you don't swab, you just get a cup full of urine, either by catheter or by clean catch, where you clean the area first, the external skin, because there's lots of microbes externally on the skin outside the urethra, so you typically want to try not to catch any of those. Usually you ask the patient to pee for a second, to like sort of clear out any of those microbes that are just sort of hanging around the surface or the external area of the urethra, and then you try to get a catch of urine from the middle of the stream. That's what a clean catch is. You can get swabs from skin lesions, where there's like a rash or a wound or something that's infected, and you can also take samples from blood or cerebrospinal fluid, which would involve a syringe, and aspiration that way. So making sure that the skin is properly cleaned in sort of all of these kind of situations, because you don't want to be getting any normal, you want to minimize the amount of normal flora that are naturally on the skin and make sure you're getting just sampling the microbes that are potentially causing the infection in that area. Sometimes lab results will come back unclear and you'll need to get another sample. So sampling technique is really important. You also want to make sure that you're doing it aseptically and not contaminating the sample with any microbes from yourself, because that can also skew the results. So patient sampling using aseptic technique is very important first step in identifying a pathogen that's causing an infection. So this is just a slide, an image slide showing you some of these different sample sites and how you would collect the samples. So for the nasopharyngeal, you could get a swab. You could also get a collection of saliva or sputum from someone, like coughing up sputum, and just you collect that in a cup. You can also get fecal samples. I'm just highlighting the ones we didn't talk about on the previous slide. Stool samples can be used for assessment of gastrointestinal infections. A vaginal swab can be done, which is done in gynecological exams a lot, in testing for certain STDs. And biopsies can also be looked at. This is just an example of an order form for microbiological testing. So just to look through it, so the top portion would be filled out by the person who took the sample and is ordering the test. So usually the doctor who's ordering the test. And it says what the source of the specimen is and what the tests are that are requested. Based on clinical presentation, the doctor will already have an idea of what is causing the infection and what tests they want to be done. Then the bottom part under the green line is all for the lab to fill out. So they can fill out results. There's a section for them to fill out results about gram stain. Is it gram positive or gram negative? Coccus or bacillus? Is it a yeast? So a fungus. And then they can do some other types of testing as well. You can do parasite testing, looking for eggs or parasites in a stool sample. So these are some really common tests that are done on microbiological samples. They even also have some miscellaneous results. So like if there's no growth or if they only get normal flora, these are things they'll check off and send back to the doctor and basically say, we might need another sample. This one didn't work out. And then they can also check off some of the genuses, any genuses that are positive in the culture results. And then down here, the bottom, is results for antibiotic sensitivity testing, like the Kirby-Bauer test, where they can use the little discs and check for antibiotic resistance or sensitivity and mark those so that the doctor knows which drugs they can choose from for treatment. So it's a form where it's kind of simplified for like maximum clarity of results. Now, for some of these phenotypic methods once we get down to the lab. So some of these examinations that they're doing, they could be doing microscopic examinations of fresh smears, of like a blood smear, for example. If somebody gets a blood test, they could take a drop of that blood, put it on a slide, and just physically look at what's going on under the slide. Are there any can they see any bacteria? They need to stain it, of course. You can also do this with a throat swab. You can stain it and look for bacteria. But also, a lot of times, in addition to looking at it under the microscope, they will also swab some agar in culture and try to grow the bacteria. Just even as a backup method in case they can't see anything on the slide, if there's too few specimens on there, etc. Biochemical testing is another type of phenotypic method. You're just looking at the traits of that bacteria. And some very common ones that I mentioned before already are looking at carbohydrate fermentation, usually through production of acid and changing color of the media, like we did with our MSA and EMB plates. Or you can do it using liquid culture and looking for gas production, kind of like what we did with the fermentation with the yeast experiment. So that was another method of looking at fermentation. There's also different tests to look for different enzyme function. A catalase test is really common using hydrogen peroxide. You just basically put a little bacteria on a slide, add some hydrogen peroxide, and if it bubbles and produces gas, that means that they make catalase. Some bacteria are catalase positive and some are not. And so you can generally observe these results either by the formation of gas or by some color change. All right, this is another version of that intero tube I showed you on the previous slide, where basically you have some tube that you inoculate the whole thing, and then you look at, and there's just these very small chambers, and you can observe either color changes or in some cases gas bubble production, and then you record the results. You score them based on a scoring guide, and then there's sort of like this method, this algorithm to calculate a code from those results, and then you can look up which organism has that code, and it's really kind of a cool way to get a diagnosis. But those things are pretty expensive. So what they're based on is basically these dichotomous keys, these flow charts that allow you to identify bacteria through different characteristics as you test them. And you guys actually you filled out one of these dichotomous keys in a lab earlier this semester. You sort of had to fill in the blanks. But so what it would look like is, you would do maybe do a gram stain. All right, look at the bacteria under the microscope. You would see that they are round, and then you determine, are they pink or are they blue? Okay, they're blue, so they're gram positive, so you're going to go this way down the flow chart. Then maybe you'll do a catalase test next. And if it's catalase negative, then we probably are dealing with a streptococcus. If it's catalase positive, then you might do another test to look at whether it's anaerobic or aerobic, and that would give you ideas of what organism you're working with. So it's a sort of a flow chart that you can use as you're doing these phenotypic observations to determine what test you need to do next in order to identify what the organism is. And we just call those flow charts dichotomous keys. Another technique that can be used is to identify bacteria through phage typing. Now remember, bacteriophages are viruses that infect bacteria, and they are super specific for different bacteria, even different strains of the same species of bacteria. And so you can use phages that have known specificities, like that there's 20 different strains of E. coli, and you have 20 different phages that infect each specific strain, and you want to figure out which strain of E. coli you have. You plate that E. coli all over a petri dish, and then you sort of grid out the petri dish, and you put a little drop of phage culture in each spot. And if you get a plaque, so 52A and 79, both result in a plaque, which means that the phages attacked the bacteria and killed them in that spot. Whereas all these other spots where there are no plaques or sort of holes in the lawn means that the phage did not work on those phages didn't work on the bacteria. So it's called phage typing, figuring out what the bacteria is using known phages that infect that type of bacteria. You can also sometimes do identification through antimicrobial sensitivity tests. There are some microbes that are naturally resistant to certain antibiotics or sensitive to certain antibiotics, and that can be used as a test for which microbe it is. And then you can also do identification using tissue culture or animal culture. This might be necessary for certain viruses that can't grow otherwise. They need cells to grow. There's also some bacteria that need cells, tissue culture to grow. They can't be grown like on agar media. So that's when we would use those situations. Now, just because you get growth on an agar doesn't necessarily mean that that organism is causing the disease. All right. So how do lab technicians know, is this really the true pathogen, or is it some contaminant? Because a lot of microbes that cause disease are normal flora. E. coli, Staphylococcus, Streptococcus. These are things that are normally found on the body. So how do you know it's not just like a contaminant and not, or if it's actually causing a disease? So there's, some sleuthing involved here. So for example, E. coli can cause urinary tract infections. It's a common cause of urinary tract infections. Just normal E. coli from the gastrointestinal tract, basically fecal matter contaminating the urethra and then getting E. coli in there. So if you have just a couple of colonies of E. coli in a urine sample, that could be contamination from E. coli that was just, in that region, like the perianal region, that contaminated the urine sample. All right, but if there's a lot of E. coli colonies, that seems pretty clear that the urine is contaminated with E. coli. So, sometimes it's not just like a clear black or white answer. Sometimes there's some gray and some critical thinking involved. So if it's a normal flora that can be pathogenic, a couple of colonies may not indicate an infection. But if it's a true pathogen, like Mycobacterium tuberculosis, which is not normally found in a healthy body, if you get even one colony of that growing, then that's a positive result, because that pathogen, that particular microbe, has no business being on a healthy person. So finding even one colony of a true pathogen is evidence that a person tests positive, whereas one colony of E. coli is not a very good, not necessarily a positive test. Repeated sampling might need to be done to confirm cultures. If you see it repeatedly, like if you keep testing that person's urine and they continually have a few colonies of E. coli, well then maybe it isn't contamination. Maybe they just have a very light infection. So if you're repeatedly getting the same results, even if they're light, that's probably indicative of a real agent of disease. All right, so that is basically everything about phenotypic examination and diagnosis. Now for these genotypic methods, and I do have a couple of videos to show you that sort of illustrate or animate the process. So we've talked about PCR before. It stands for polymerase chain reaction. It's basically doing DNA replication in a little tube. So you add nucleotides and some template DNA and some DNA polymerase, and you incubate it at different temperatures, and you get lots and lots and lots of copies of that original piece of DNA. And in the case of diagnosis, we use primers for a suspected microbe, and if if the primers match the DNA, then you get lots and lots of copies and you have a match. But if they don't match the DNA, then you don't get any copies, and you know that that's not the microbe that's causing the disease. So you do have to have, with PCR, it's not sequencing, it's just making copies and using primers that you've already designed to a specific microbe. So you kind of have to have an idea of what the pathogen is to do a PCR test. Like you're testing if this person has flu or HIV, and then you get a positive or negative result. It's not DNA sequencing, it's just making copies, and you only get copies if it's positive for that organism. So this is just a quick video of how PCR works. The music's kind of dramatic, and I may have shown this in class already. The first step of PCR is going to be melting the strands apart, which we call denaturation. I'm using heat to melt the two strands of DNA apart. And then we have the annealing stage, where the primers come in and find their complementary sequence. There's the annealing. And now the extension stage is where the DNA polymerase binds to the primer and starts synthesizing a new strand of DNA, of that sequence right where the primer has bound to. And then we start again. We melt the strands. We anneal the primers. We do extension, make new strands using the DNA polymerase enzymes. And every time we do this, we double the amount of DNA in the tube. And so when you double and double and double and double, you get exponential growth. So this nicely shows that exponential growth after 4 cycles. Right, after 20 cycles, we already had 1 million. And after 30 cycles, you have 1 billion. More than 1 billion pieces of that DNA. And then you can easily visualize that on gel electrophoresis, or sometimes you can add some fluorescent indicator and then say you have a positive test. Magic of PCR. Okay, so that's one way to do a test of using DNA of a pathogen. Another one is to do a hybridization test, and this one uses probes. So basically you add a spot of the, like let's say you take the swab and you put it on a piece of filter paper. You just dab it on a piece of filter paper. And then you can heat the filter paper to lyse the cells and expose the DNA on that filter paper. And then you can add DNA probes that have a fluorescent tag, and you can look for that fluorescence. And again, a quick video on this technique.
Bacterial cells containing specific DNA clones are identified by a technique known as colony hybridization. Cells from each colony are first transferred to a filter. Treatment of the filter with denaturing solutions lyses the cells and releases the DNA. The denatured DNA adheres to the filter. The filter is placed in a bag with labeled probe to allow hybridization to occur.
And the probe is some known DNA, so DNA for, E. coli or flu.
Washing the filter with fresh solution removes excess probe molecules that are not specifically bound to DNA on the filter. Emissions from the labeled probe create black spots on X-ray film. Comparison of the location of the probe signal on the film with the location of colonies on the original agar plate identifies colonies containing DNA complementary to the probe.
So you can do this with a whole plate of bacteria, or you could do it with simply the patient's sample. You could just dab it onto the filter paper and test that one spot. So there's different ways to do this hybridization, and I'll show you on the next slide what that looks like. Ribotyping is DNA sequencing, and it's sequencing of a specific gene for ribosomal RNA. And the reason we use that gene is because it's highly conserved amongst different organisms, and so it doesn't change a lot. So it's, a lot of genes, we, as humans, we all have different phenotypes because we have different genes for like skin color and hair color and eye color, and there's all kinds of different genes, like different variations of genes that we have, but we all have the same gene for ribosomal RNA. That one does not change or vary between different individuals of the same species. So it's a highly conserved segment of DNA that's used to identify a species of organism. This is a slide just showing you what hybridization looks like. So you could take colonies from a plate or just directly from a swab sample, and you swab it onto a spot on filter paper, and then you add some kind of solution to lyse the cells and stick the DNA, get the DNA stuck to that filter paper, and then you add a specific probe. So maybe to one spot you add a probe for E. coli, and another spot you add a probe for Salmonella. And then you, these probes are just little pieces of, they're basically DNA primers, but they have a little attachment to them. They're fused, chemically fused to either an enzyme or a fluorescent molecule or a radioactive molecule, something that's going to allow visualization in the end. So after you bind the probe and then wash off any probe that didn't stick, you then maybe add some component that the enzyme will cleave and make it change colors, or you look at it under a fluorescent light to see if it fluoresces, or you put it up against some film to see if it's radioactive, depending on what that probe molecule is tagged with. So in this one, it looks like it's tagged with an enzyme that stimulates a color change when you add a certain substance. And so this one changed color, which means it's positive for, I guess we said Salmonella was this probe, but it's not positive for E. coli because that one didn't change color. So hybridization usually involves it's a visualization of DNA using this sort of trick of hybridizing, combining the DNA probe or the primer DNA with something that is visual, like a radioactive group or a fluorescent group. Another way that we can look at DNA is we can basically do DNA fingerprinting. So we've talked about gel electrophoresis before, where you can do DNA fingerprinting, where you take the DNA of a microbe and you use restriction enzymes to cut it up, and then you run it on this gel using a electropulse, using an electric field that pulls the DNA through this gel matrix, and the shorter pieces move faster and the longer pieces move slower, and so then they separate into this sort of banding pattern that you can see in the gel. Well, this is the same thing, except it's a little bit fancier. It uses multiple electric fields to pull the DNA pieces not just into straight lines, but sometimes into crazier patterns that are just even more separated, so you can get a better picture, I guess a better fingerprint. But it's essentially the same thing. It's taking DNA, taking a bacterial culture, mixing it with, treating it with some kind of enzymes to cut it up, and then loading it into the wells, these little divots in the gel, in the agarose gel, which is like a slab of jello, and then putting it in a machine that sends electrical impulses through it, and that pulls the pieces of the DNA apart at different speeds, and you get this banding pattern that can be read, and it's like a DNA fingerprint of a microbe. All right, so those are the genetic techniques. Now for the immunologic techniques. All right, we call immunological testing serology. And we do serology using antibodies. We can either use known antibodies, which is what we're doing on the right side here, where we take the patient's sample, we take the microbes from the patient, and we use known antibodies to a known pathogen, and we see whether that's a match. So for example, if we want to know whether the patient has Staph aureus, we can take a sample of their bacteria and mix it with antibodies that we have that are against Staph aureus, and if the antibodies match, then they cause all of the cells to clump or agglutinate together, which is the same thing they'll do in the body. But we can't see bacteria that are spread homogeneously in solution. Maybe it just looks like kind of cloudy, but when the antibodies are there and they cause the bacteria to clump, you can visually see these clumps that form, and that's a positive test. The other thing we can do is if we can't culture the patient's microbes, what we can do is we can take antibodies from their blood and test it against known samples of a microbe or antigen. So we can take a culture from a company of Staph aureus, or we could order just proteins, antigens of Staph aureus, and mix them together and see if they are a match for the patient's antibodies. So it's sort of two different, you can use it to test the patient's antibodies using known antigen, or you can use it to test the patient's antigens, the microbes, using known antibodies. But both of these are, they're very simple, very quick, rapid tests, and they're very sensitive. Antibodies are very sensitive for microbes, so even a small number of antibodies can cause this agglutination or clumping. So we can visualize these using these agglutination or precipitation tests. They're virtually the same thing. In an agglutination test, you're looking for clumping of whole cells, whereas in a precipitation test, you're just using antigen. You're testing antigen, so it's just protein, not whole cells. But either way, they're both virtually the same test. You are using antibody and antigen, and if they are a match, then they'll clump together and you'll get this visual, you'll be able to see this sort of precipitate or clumping. And it's due to antibodies recognizing antigen or cells and causing them to clump together because they're all cross-linking. We use this a lot for salmonella testing and for strep testing. And then lastly, some different antibody tests that we can use. We can test a patient's antibodies for a specific organism by taking titers, which is a way to measure the amount of antibodies they're producing. This is commonly done in pregnant women. So rubella is a virus that is teratogenic, and causes birth defects, and a pregnant woman who gets rubella is in danger of miscarriage or serious fetal defects. And most people are vaccinated with the MMR as a child, and so they should have protection and immune memory and making antibodies towards rubella. But when a woman gets pregnant, one of the first things they'll do is order a blood test to test her titer of rubella antibodies and make sure that she is still immune to rubella. And if not, if she doesn't have good titers, then they will probably re-immunize her, give her another MMR. Some people just don't seroconvert, they just don't make antibodies. And in that case, then the doctor would be just much more I guess, aware and look out for any symptoms of rubella in this patient and really maybe encourage them to social distance a little bit, especially from kids and people who are unvaccinated with MMR. So titer is just a word that means sort of like measuring the concentration of antibodies that you have, either total or towards a specific pathogen. A Western blot is a technique to look for proteins. It's sort of like a DNA fingerprint, except it's a protein fingerprint. Similarly, you run proteins out on a gel, and then you get this banding pattern that can be visualized. And this is used, this is a common test that's used in diagnosing HIV for viruses, looking for viral proteins, because we can't culture viruses, so immunologic assays are often what's used for testing viruses. I'm going to skip over immunofluorescence. That one's just visualizing using fluorescence. The lateral flow immunoassays are these really quick use kits. Most people are familiar with them as pregnancy tests, but we also use them as rapid strep tests and rapid flu tests and rapid HIV testing. So these are little assays that have been made using antibodies towards a specific antigen. In the case of pregnancy, the antigen is HCG. That's what you're trying to detect. And so what happens is you have this sample pad. So the spot where you add the sample, all right, has some little beads basically with antibodies on them. And if the antigen is in your sample, whether it be HCG because you're pregnant or flu because you have the flu, all right, those beads then move via capillary action. The water just pulls them, the liquid pulls them up this filter strip and to the test zone. If you have antigen, then those beads will stop there and stick to the antibodies in the test zone, and these beads are colored, so they'll leave this colored strip. And then there's also always a positive control test strip, or a positive control strip, that binds to the antibody portion. So whether or not there's antigen, this strip will always light up. So a negative test has the control strip light up. So usually you have one line on a negative test, but you get two lines on a positive test, whether it be for flu or strep or pregnancy. They all work that same way. One line is just the control strip. It means that the test works. It's not a dud, it's not defective. But two lines means that you actually had a positive result. And you can actually order these rapid strep test kits online. You can get them on Amazon, which is what I usually do for a strep test lab that we do, because they're so commonly performed in lab offices that I like for you guys to perform them. But don't worry about ordering them yourselves, but know that you can if you ever wanted to. I know people who have ordered them off Amazon because they get strep throat a lot, or their kids get strep throat a lot, so it saves them a trip to the doctor's office to be able to sort of self-perform, but it still involves throat swabbing, which can be done wrong, so I don't generally encourage people to do it themselves. All right, and then another one that's really important to know about are these ELISAs, or enzyme-linked immunosorbent assays. I just have to check the time. Okay, so I'm going to have to come back and make another video about this because I have an appointment to get to. So we'll make one short video more about ELISAs coming right up.